this is the last session of this beautiful event. So the next speaker will be Olivier TABOUREAU. He has a background in biochemistry and obtained his PhD in 2009 in bioinformatics and data mining. Then he moved to industry, to Denmark, where he studied antimicrobial peptides and coagulation factors using QSR modeling and molecular modeling approaches. In 2005, he joined the Center of Biological Sequence Analysis at Technical University of Denmark, where he created a group of computational chemical biology. In 2012, he came back to France, obtained the professor at the University of Paris Diderot, where he started work on computational pharmacological profile and created software web server ChemProt to mine chemical biology data. And since 2018, he is a member of the computational modeling of protein ligand interaction team at the biological and functional unit at the Université de Paris Cité. With his expertise in bio and cheminformatics, he has contributed and developed to the development of computational approaches related to pharmacology, drug safety, and chemical risk assessment. He also has such particular interest in graph theory, network biology, and systems of pharmacology analysis. So, please.
Very much. I can see my resume was quite long, so maybe I don't have anything else to say now. A part of network science. So yeah, this talk will be on chemoinformatics and network science and how we can use network science with some chemoinformatic tools. Before to start, I will just present rapidly our team and what we are doing. So as it was mentioned, I mean, now we are on a team which is called Computational Modeling of Protein Ligand Interaction at the University of Paris Cité. And the main objective is really to understand the relationship between small molecules. It can be any kind of molecules, so drugs, environmental chemicals, natural products, peptide, to see the connection with proteins and genes. And then with that, to make a relationship with disease, because I mean, in many cases, we have information about compounds, we have information about disease, or maybe some phenotype, but then we don't know the information which is between. And for that, so we have some different area of expertise, people working on molecular modeling, do some docking, some dynamic simulation. And that will help to do what we call some multi-target profiling or pharmacological profiling. And the second part will be also more related to integration of data and biological data and essentially genomics data, because now we can have access on such information. And that's help also to enrich, I mean, this pharmacological profiling, basically, I mean, to enrich, I mean, this heat map, where, I mean, we have a lot of green color, meaning we don't have any information between connection between small molecules and proteins, and using this genomic information, which are basically some gene expression data. Now we can get more than 50,000 molecules with such information, using some pathway and protein-protein interaction, using also some genetic variation. We can more easily to do some connection between small molecules, clinical effect that can be beneficial or adverse drug effects, and so try to really understand the mechanism which is behind that could explain then, I mean, why small molecules can be beneficial or might be also generate some adverse drug reactions. And so today I will talk a little bit more about network science, because it's one area also where I'm working a lot. I thanks, I mean, Artem Cherkasov, because he presents already this slide, so don't have to do anything else on network. Apart that, if it's something that you're interested in too, you can go also to Wikipedia. You have a very nice description and definition of what is network science. It's basically, I mean, some small spheres that you connect with some others. You have a link, and then, I mean, you do a lot of statistics on that, basically. But more interestingly, that you can use such kind of information with biological data. And what is interesting in our work, in our research, that you can integrate different sources of information. You can integrate, I mean, some molecular information. We'll see some examples, but you can integrate also some cellular information, some pathway information, and make some connection from different sources of information. You can generate some statistics on different area of your biological network, of your networks, and then, I mean, reach different question and respond to this question. Probably, I mean, many of you are using some social networks, Facebook. I mean, that's for the whole people. Now the youngest one are using Instagram or Snapchat. But what is interesting on such kind of networks, there are some work that show that basically you can have 6 degrees of separation between any kind of peoples. And so that means you can be connected to Bill Gates, you can be connected to Rihanna, if you like her, I mean, or to some other actors. And it has been shown that on the social connection, you can have at the maximum 6 degrees of separation between you and some actors, famous actors. And that's the same in biology. Basically, I mean, you could integrate 6 sources of information, 6 kind of different information, and make some connection between them, and using some statistics to make relation between this part, proteins and disease, and see, I mean, what happens, why you have a relation between the disease and the small molecules through these different pathways and proteins. And such kind of networks, I mean, it could be done also with us. I mean, we could make a network on the Chemoinformatics summer schools today. See, I mean, what is the connection between the organizer? What is the connection between the speakers? What is the connection between also of the research area? People are more on QSAR, some other on molecular modeling. And then, I mean, you could cluster them. You could find, I mean, who is the central connection of that. So basically, you could make a cluster based on which area you are coming from. So you have some European mafia, you have the French mafia. I can see a lot of French people. The US mafia, the Japan mafia. I didn't put the Russian because those people are a little bit on the borderline. I mean, they are everywhere. They are in US, they are in France, they are everywhere. But what is interesting also on such kind of network that, yes, you see, I mean, maybe the hub, the cluster connection is Alexander, which is not completely true, because when you make some emails, you usually have connection with Olga. So that means, yes, you have a hub, you have, I mean, a strong central connection, which is Alexander's, but around him, you have also a lot of people, which is connected, I mean, to Alexander, but which is also connected to you. And they have some influence then also on if everything is working well or not. And that's the same also in biology. I mean, you can have a genes, a protein, which, I mean, it's the most important guy to do something. But then around, I mean, you have a lot of proteins, and Artem was talking about protein-protein interaction. That's helped you to see, I mean, what are the others' connection to this central person, and what are this other connection with the other peoples on the networks. And so like that, you can do a lot of suggestion about possible protein genes that can be connected to small molecules and to some phenotype. That's what we do, and that's what some others do also on what we call network pharmacology. So here it's an example where you can see people were working on different drugs, imatinib, the natalizumab, the tanezumab, and they were looking about what are the proteins which are shared between these different drugs, and what is connected or shared between them. That's what is in yellow. And from such kind of map, you could assume that, yeah, imatinib and the natalizumab, they have some shared proteins, so they are a similar mechanism of action. But then, if you want to treat, I mean, peoples, it might be more interesting to use, I mean, the green one, because the mechanism of action is different. So a combination of imatinib or tanezumab with the natalizumab might be a higher performance than if you use, I mean, the 2 drugs, imatinib and the tanezumab, because they are on the same mechanism of action, those one. And network pharmacology can help you to make such kind of decision of what you could combine or not. I mean, what is shared by a protein or not, and what could be related to a disease or make some report protein effort on that. That has been done also on some more general networks, drug target networks, and that's what is using also for some COVID research area, where one of the ideas was to use some repurposed drug to see, I mean, what are the proteins that has been shared between these drugs targets and the COVID targets, to see, I mean, if we can repurpose some drugs related to that. So, I mean, such kind of network science, they are applied on many, many different ways in network pharmacology, in network human disease also, on the COVID-19, and also with exposome research, where we want to see, I mean, the combination of exposition to different chemicals, how they can be related to some disease or leading to some disease. In our groups, long time ago, we were also already trying to infer some relation between protein chemical association and see from a bipartite network how we could, I mean, generate a monopartite networks that could give some suggestion about some proteins that can be connected to each other and that could explain to be related to some disease. It is based on what we call the guilt by association theory. I mean, that means if you know somebody in the jail, you have some suspicion that you might be also guilty or you have some a bad past. And so you are connected to this guy. And that's the same with protein. We can believe that if there are some drugs which have some interaction and some activity on different proteins, then, I mean, probably you have some connection between these proteins. And so when you build or design a new drugs, you might make some hypothesis that these drugs could also influence some performance on some of these proteins and so be related to some proteins. And so we have many types of biological interaction. As I said, I mean, drug proteins, protein DNA. And as I said, I mean, like the 6 degree of separation, by including all of them, you can make easier some connection between molecules, phenotypes, and the mechanism which is behind. That's what we have done, I mean, also 10 years ago, where we are associating drugs, targets, clinical outcomes, and try to see, I mean, on which compartment or which kind of tissue you could have a adverse or higher adverse outcome effect compared to a therapeutical effects. And at the end, I mean, we could see with such kind of network that drugs are more likely to cause side effect in the organ tissue where it's more likely to accumulate. And in some way you could say, yeah, it's obvious. I mean, if you are using a CNS drugs, it will have a good impact on your brain, but then it will accumulate also there, and then the side effect will be probably stronger or higher on this tissue. But then you can quantify them, and that's the difference. That was an introduction in network biology. If you want more, Abel Laszlo Barabazi, I mean, make a nice book on that. You can find a lot of information about how you can use it in different areas and what are the algorithms and statistics that can be used with that. But in our lab, and today I will more concentrate the work on using network science in toxicology and adverse drug reaction, because it is something where basically we don't have so much information about the mechanism of action. You take a drug, what you will look in preclinical or clinical, it's what is the adverse outcome and what is the beneficial effect. But then you don't know, I mean, what will be the mechanism which is behind the adverse drug reactions. And so it's a problem. It's the same with exposome research. You have the molecules, you have the phenotype, but how, I mean, you make the connection between the molecule and the phenotype, it's not so easy to predict and to give, I mean, what could be the protein or the genes. You have some few exceptions, of course. I mean, you can use a hair potassium channel, you know, it's related to some cardiac effect. But apart few of them, the connection between adverse drug reaction and compound, it's not so obvious. And so what we try to do is to make some suggestion about what could be the protein or the genes related to some adverse drug reaction. And we use, network science for that. So one thing we started to do is to use, drugs for whom we had some adverse drug reaction. So we have 1,000 drugs, more than 6,000 adverse drug reactions. This adverse drug reaction could be classified on what we call a system organ classification. And so like that, you can do some statistics, look on how many compounds are related to some adverse effects. So you can see the most common adverse drug reaction are nausea and vomiting. Nothing new also, we know that. But then you have some specific adverse drug reaction which are only related to one drug. Start to be interesting because then you can look for these drugs, what are the targets, and start to make some connection about what can be the protein related to this adverse drug reaction. And also, we have seen we have more than 155 drugs which are associated to only one adverse drug reaction. Again, it's easier to make some connection about what could be the target related to that. And as for all of these drugs, we know, at least we have some information about what are the protein targets. So we know now what is a drug adverse drug reaction association. We can make a network combining both information, which make, if you put some colors, and I put some pink colors for the ladies because I know they like that. But you have something which is very bulky and say, okay, what can I do with that? It's the same like a GPS map. It's the same as a heat map. Of course, using as it is, you will not get anything out. But then, you can research by compound, you can research by protein, you can research by adverse drug reaction, and start to make some hub and see, okay, what are the connections for this protein? How many compounds I have? How many adverse drug reaction? The same for adverse drug reaction. You can define, what are the protein connected? What are the drugs? And if you focus on specific question, start to suggest, what could be the protein related to that? Doing that, we could see there were some targets associated with the most ADR are essentially cytochrome and transporter. Also, something that we were expected. We know that the metabolism enzyme, are highly related to some adverse drug reaction. But we know also for some of them, we have some specific role of protein for specific ADRs. That's the case for HPAD, which is a hydroxyphenyl pyruvate deoxygenase, which seems to be highly related and specifically related to hepatocellular carcinoma. Okay, that's good. Something I didn't know. The same for SQLE, the swollen expositase. We can see with such kind of information, it's connected to a blister, and it seems that it's one of the unique protein related to that. Good. So we have some specific response for some specific adverse drug reaction. But then for many of them, majority of protein, we don't have such kind of specific relationship. So then we implement a score, basic score, I will say, where basically according of the number of drugs related to adverse drug reaction, the number of protein targeted by this list of compound and related to the adverse drug reaction, you can make a score that defines the importance or the contribution of each protein to an adverse drug reaction. So then, that means for one adverse drug reaction, like the prostate cancer stage 4, you have only one drug. So, okay, it might be not so optimal to see the contribution, but you can see and say, okay, I know I have the gonadotropin receptor and the cytochrome 3A4 that seems to be related more specifically on this prostate cancer stage 4. But then for some other, like the bone fragmentation, the list of proteins, it's longer because you have more compounds. You have 4 compounds which are related to this bone fragmentation. But then you can see some of the genes seems to be more related to this bone fragmentation because these 4 drugs, all of them has been related to the FDPS proteins. So probably they have a higher influence, higher contribution to this adverse drug reaction compared to the other genes where it's only 1 compound or 2 compounds which is related to that. So then for each adverse drug reaction, you can suggest, define what could be the mechanism of action, what could be the protein and the contribution of this protein to an adverse drug reaction. Of course, it's depending of the drug targets and the drug protein information you have. Actually, for this study, we use only drug bank information and I forgot the other one, drugs.com, I think, information. But we could enrich this information with what you can see in Campbell, what you can see in PubChem. What is important is to be sure that the protein you include on such kind of network, you know you have a strong activity of the compound on this protein. Otherwise, you will start to have some false positive. And then, based on that, you can go further. You have information about drug, targets, adverse drug reaction. You can also now see what adverse drug reaction could contribute to another adverse drug reaction. It's like symptoms. You have vomiting, you don't feel bad. Is it something related to the drugs and vomiting, or is it maybe something that you had previously, which was nausea? So then you can try to make some relationship between adverse drug reaction and see, I mean, if one adverse drug reaction will lead to another one more seriously, with more problems, and see, I mean, how you could relate such kind of information still rated by compounds. So that's what we did. So we still have our 1,000 drugs, we have our 6,000 adverse drug reaction, and depending on the number of compounds that are associated to this adverse drug reaction, you can start to infer and, let's say, I mean, transform from a bipartite network to a monopartite network. What is the connection between the adverse drug reaction? So some of them are also obvious. We can see that the loss of consciousness, syncope, headaches, related to diseaseness seems to be okay. But you have some other which are, I mean, more surprising, but could be also related, like rash, which is also related to the diarrhea, like pyrexia, which is also related to anemia or neutropenia, like respiratory arrest, which is also related to overdose, or cardiac respiratory arrest. So like that, you can make some hypotheses about, be careful, an adverse drug reaction could lead to another one. And we have seen the size of the edge can give also, I mean, the contribution, I mean, the importance of the relation between these two information. And that only using drug, targets, and the phenotype information. So when you can apply such kind of thing in one area, you can do in several other areas. And we continue also such kind of research with network science on environmental disease network. The principle is the same. We have some chemical information related to disease association. And then from that, we try to predict what could be a disease leading to another disease, doing some disease association. And what we try on this work, we try to make a kind of semi-quantitative, because for each compound disease association, we are in a curve of degree of evidence. For some compounds, we knew that it was leading to some disease. For some other compounds, the evidence, the epidemiological evidence was not so clear, and for some other, it was quite limited. But then, I mean, you could develop 3 different networks, make a score of evidence on these 3 evidence network, and then build a unique one where, according still of the size of the edge, you can define, I mean, what is the importance of one disease related to another disease. And again, I mean, that's what you see to make relation between disease and disease. But then you can go back, and as you have some information about chemical and protein information, try to do some prediction. What could be a list of genes or a list of proteins that you know the compounds will have an effect, and then can be related to this disease association. So you can really go from molecular evidence to some phenotypic evidence and make connection and give some explanation about the mechanism of action that could explain a relation between a small molecules and a phenotype. We did the same work also with some toxicity information and tissue information, where we have a list of 25,000 chemicals. We know, I mean, on which system they could have an impact. By system, it means tissue. I mean, you have the reproductive system, the cardiovascular system, the liver system. And we knew that these chemicals they had some toxicity effect on these systems. And for these chemicals, we have information also on proteins, and the proteins we knew if they were expressed on some tissue or not, so you can clean the data. And so like that, you can develop a model, and that's what we have done about compounds, protein, system related, and so make some prediction about what could be the proteins that are impacted by chemicals, and that could, lead to some toxicity on different systems. One thing that we are working on right now, it's to try to quantify this importance, because that's one of the limitations we had right now on network science. You have two spheres, you have a link, and the contribution of the links between these two spheres is usually based on qualitative data. And one of the questions is when you do some inference, meaning that when you have a bipartite network and you want to transform in a monopartite networks, how much information you lose and how much then the evidence you have between two events. And so we tried to use an algorithm which is based on the work from Vogt in 2019, where basically they are using the Shannon entropy concept that measure, how many associations between drugs and events are spread over all the possible connections in the networks. And so doing that on the bipartite network and on the monopartite network, you can estimate how much information you lose between these two connections that you have at the end compared to what you have at the beginning. And so here it's only an example, but basically according of the color you have between the edge, color edge between two spheres, you can define, how much loss of coverage you have when you make this transformation. So the closer to yellow is lower the loss of information. You have a strong connection between these two. The higher to black, the more information you are losing when you are making such kind of transformation. And so it gives like that a possibility to, let's say, give more importance for some connection, some protein to disease or ligand to disease, compared to some other where you say, I don't have any strong information, strong connection, so probably the contribution is lower compared to the rest. And so doing that, just checking the time, I'm fine. Again, you can go back to the compounds. You can comment on any kind of information related to these compounds. You can see also how many drugs that share similar connection with some of these events. And then when you make the transformation, define, what are the strong connection related to this compound and what are the lower contribution for this compound. Finally, I will go to another topic, which is basically now we can enrich, and that's what I have shown to you with this network science, different type of information, compounds, protein, disease, pathway. And since few years now, we can get also information about morphological cell change that you can get from the cell painting data. And Olaf was talking about that yesterday a little. What is interesting that here you can estimate and measure the change of the cell. So then you can include this information as well and see what could be the relation between the perturbation at the molecular level on the protein or on the gene, and how you could relate this information to the perturbation at the cell level. And at the cell level, what you have, it's an image. So it's different image like that, when you can have different parameters, which can be related to the shape, the texture, the intensity. And so give some explanation. Okay, be careful. If you are making some perturbation of this protein, you might also make some perturbation on the cell morphology. And if you make some perturbation on the cell morphology, you will probably also generate some trouble on some phenotype and maybe some disease. And so now we started to include also this information. So now we develop a kind of platform, which is in Neo4j, where we can connect, compounds based on the structure. So we have a scaffold also of the molecule, different descriptor of the molecule, where you can connect, the target, you can connect, disease association, pathway association, but you can connect also some cellular perturbation on these networks. And so try to have another set of information that can explain or give some explanation about the relation between compounds and phenotype. So yeah, for compounds, you can get information about proteins targets that are perturbed, fine. But then you can get information also and make some hypotheses. We give a p-value to have some confidence also of what we get with the compounds and some different information and disease information and cellular morphology perturbation. And the next step on which we are working now, so we link, I will say, the proteome perturbation to a cell perturbation. We are looking also now with transcriptomics data perturbation, gene expression deregulation. And we are trying to integrate such information to have also another layer of relation which will be not related only to protein, but also to the gene deregulation. And so like that, you can, for compounds, define, here's an example, how many genes are perturbed, what are the diseases that can be related to this perturbation, and also what are the cell morphological cell perturbation that you can see with some p-value. So it gives also some confidence, but is it something very important or not? So in conclusion, the idea of using network science that you can really put different building blocks of information and connect them with some edge, give some importance with this edge and with some statistics about the contribution of different partners on the proteins, on the genes, on the cells, to explain, what can be the relation about the molecules and the phenotypes. And the chance that we have right now that we can get information on such kind of omics information, if I can say that, on the different layer, on the molecular interaction, on the cellular response, on the organ response, and also at the population levels. One thing also we are starting to consider now, it's that the genetic variation, because we have such kind of information available, and we start to look also how we could explain, how a SNPs, or maybe also how a full genes which are not present could be related to less or lack of efficacy for drugs, and so give some explanation about pharmacogenomics for drugs. To finish, I would like to present some of the people who are working with me, actually, people from, that's essentially, PhD and postdoc who are working on such kind of integration. And also, you have seen probably yesterday evening, Karine Oduz and one of our PhD who are working also a lot on network science and that have contributed also on this work. And that's also some of the partners and the EU project on which we are working. And I'm ready for questions.
So we have a few minutes for a few questions. Are there somebody? Yes, please.
Thanks. It's a very nice talk. I have one question, especially for the ADRs and essentially things that you get from the clinics. How can you integrate into these things now? Let's say comorbidities that patients typically have. So you see an ADR, but there is some state with that patient that actually had the ADR. And the same is true, co-administration of other drugs, because one drug might influence the other one. Is there any way to do this, or any plans to integrate these kind of things?
So it's something we, right now, as you said, it's essentially, public available, general information you can get. But then, we are working also with some collaborators in Denmark, in Copenhagen. I know some of the people there, and they have also access to some patient information, patient records. And so we start to look also, then as you said, exactly, how patients are using, maybe a cocktail of drugs and how they can be related to some adverse drug reaction, to try to optimize such kind of network and say, okay, we know now it's not only this drug, but that will be a combination of 2, 3, or maybe 4 drugs that explain, I mean, or will have a higher influence to lead to such adverse drug reaction. And yes, I mean, that's such kind of information we need to be more precise and more specific on what will be the real mechanism of actions that relate, I mean, an adverse drug reaction to a compound.
Other questions? Yeah, if not, we can. Thanks, Olivier.